Today we'll talk about the basics of WHO classification 2016, and we expected that it will be changed in later this year, 2021. Anyway, 2016 WHO classifications, to explain, you have to know that it's a combination of genotype, the genetic mutations and molecular information that we know now about the tumors, adding to the phenotype, which is how the tumor looks under microscope, including the cell type and the characteristic of mitosis or atypia, leading to this classification 2016. If we go to the phenotype and trying to understand the cells of the CNS, mainly is neurons and glia. So neurons is the main neural cells with the main functions of transmission of impulses and store information. On the other hand, we have glia. Glia is supportive tissue to the CNS. We have astrocyte, which is mainly supporting other connective tissue of the CNS and also playing a part in the blood-brain barrier. We have also oligodendroglioblastoma, specialized epithelium lining the CSF, and oligodendrocyte, which is mainly for myelination, and microglia, which is the macrophages in the CNS. So the phenotype, if we look under microscope, we can see the star-shaped astrocyte here, and we can also see the epidendymal cells, the cuboidal cells lining the CSF spaces, and we can see oligodendrocyte, which is having a what we called characteristic prenuclear halo surrounding the nucleus, and this will play a part of how it's appeared later on. There's things to differentiate between the CNS tissues and maybe metastasis. For example, metastasis called GFAP. It's the glial fibrillar acidic protein, which is most of the astrocyte of the CNS tissue, including astrocyte, odendroglioma, and also ependymoma, can be easily suppressed by them, and also S100 protein. Also, not only the cell type is important, it's also the WHO grading, which is amine, which is the characteristic of the mitosis and proliferation. As we know, amine is a mnemonic for atypia inside the nucleus, and M for mitotic activity, and including anaplasia, and then E for endovascular microvascular proliferation, and N for necrosis. If we can see the grade 2 and 3 and 4, the difference is the microvascular proliferation and necrosis is only present in the grade 4, but the mitosis is present only in grade 3 and 4. And just atypia will have a grade 1 or 2. So, some people say, if you can see mitosis, as you know, mitosis is a continuous process that traditionally divided into 5 phases, and it's very important in physiology of the tumor that he has a very high rate of proliferation. And they say if you can recognize 5 mitosis in high 10 in 10 high in high power high in 10 high field in 10 power high field magnifications, that means that this patient had a very tumor. It considered tumoral, but they trying to invent some quantitative, semi-quantitative score, which is MIB-1 or Ki-67, which is an antigen expressed in all phases of the cell cycle except in G0. It's a valuable marker for cell proliferation, but only can be used in the fresh frozen specimen. And as I told, MIB is a monoclonal antibodies against Ki-67, and we can see how much can use to compute the quantitative. So if it's more than 7 to 9%, this is indicative of anaplastic tumor. So finally, we can see that WHO classification 2016, it's about integration between 3 layers. So layer 1 is the histological diagnosis of the cell type, and then WHO grades reflecting the natural history and the rate of the proliferation, and at the end, molecular information. For example, diffuse astrocytoma IDH mutant grade 2. That means that cell type is this astrocytoma under the microscope. This is the phenotype. This is the WHO grade is 2. That means that she has an atypia, and but no necrosis or endovascular proliferation, and high mitosis, and IDH mutant meant that the genes for IDH is mutant, which we'll learn more in the next videos.